Assalamu alaikum everyone. My name is Ikra Zahra and today I am going to talk about my topic of presentation, which is chromatography. So our experiment is experiment number 10 to identify unknown amino acids by paper chromatography. So here is the slide in which we have mentioned the name of our group members and their roll number. Then we have mentioned all the topics we are discussing in the coming slides. So our first topic is introduction to chromatography. So at first we have to know that what basically the chromatography means, or the definition of chromatography. That is, the chromatography is a separation techniques based on different relationships of compounds. So basically it is a laboratory technique for rapid and efficient separation of mixture, or might be the purification of any compound. Now the differential migration of various components of mixture is through two phases, namely the stationary phase, which is under the influence of a mobile phase, which is also called a moving phase. So we will discuss these two phases briefly in the next coming slides. Here just keep in on mind that the mixture is dissolved in a fluid, which we call it a mobile phase, or that dissolved fluid is carried out through a system on which is fixed, or a material phase, or a stationary phase. So now we are able to figure out the definition or intro of chromatography, which is basically it involves passing mixture in a solution or suspension through a media in which the component flow at different speeds depending upon the which substance it is, or in other words it is a method for separating or identifying the particular component of mixture, or it basically a method in which we allow the molecules of mixture to spread themselves between two medium, that is the stationary or the mobile medium, which we have already overviewed. Now coming towards the importance and application of chromatography. First we will discuss the importance and then the application. So importance of chromatography. So the first importance is it serves as an important analytical tool. Basically why we are saying it as important tool, because it is a tool for purification and isolation of compounds from the complex mixture or their proportion. Why we are saying it analytical, because it is a method for the qualitative or the quantitative analysis of a component of a mixture. The second importance is not only dissolve but also isolate or partially describe the different compounds, which we say that it serve as a mean for the resolution of mixture, or for the isolation or partial description of separated substances, or it also allow the separation or partial explanation of unknown chemicals or substances, unknown unexpected chemicals. So now the last but not the least importance of chromatography is it is an essential laboratory approach in any science that deals with the chemical components or reactions, or deal with any process. So that we can say that it is the most selective or most widely applicable technique for the separation or separatory technique. Now coming towards the application of chromatography. So it is applicable for separating mixture of compounds that might be amino acids or proteins or carbohydrate. It is also used for the analysis of drugs or hormones or vitamins. For example, if anyone has consumed nicotine in his diet, or in smoking or any other stuff, often in a small amount, so it will be absorbed in blood. So we can easily analyze it in our blood or urine or any other body fluid. Or it helps to identify unknown compounds. We have two amino acids, we can identify any one of them through this technique or separating the compound mixture. Or it help us to determine the purity or concentration of chemical in the pharmaceutical or biotechnological sectors, or stuff like that. Or there are many more applications of chromatography.